Okay, so this is a super up close and personal view of part of a red blood cell. And inside the red blood cell on the right hand side, we've got lots and lots of these red sort of, they almost look like four-leaf clovers without a stem. And those are hemoglobin molecules. Hemoglobin's the protein that carries oxygen. We've got some cytoskeletal proteins, and then we have proteins that protrude through the membrane. And these are actually what are called glycoproteins. So a protein that also has sugar groups on it. And that's actually what determines blood type. We're going to wait and talk about blood type when we do the immune system, because until you understand something about antibodies and antigens, which is the territory of the immune system, understanding why being transfused with an incompatible blood type can be life-threatening doesn't make any real sense. Students end up being confused, and then they kind of just turn off. And if you're going to be in health professions, it's one of those things that you really just have to have in your head. The rest of the screen represents plasma, which is the ground substance of blood. So we're going to talk about the functions of blood, what blood is made of, how oxygen and actually carbon dioxide are transported, and then a little bit about blood clotting, which involves platelets, hence the dorky science joke. Okay, so blood is sort of a liquid highway. Its primary function is to transport oxygen, nutrients, and hormones to our cells, and to carry metabolic waste away from cells. So metabolic waste includes carbon dioxide, CO2, which is disposed of by our lungs, and then nitrogen-containing waste, which is removed by the kidneys. So when we talk about transport of waste, I always try to say metabolic waste, but waste is really the job, getting rid of it is the job of the urinary system and the respiratory system. And the digestive system is kind of disposing of indigestible parts of our food. Blood also contains substances that allow for the components of blood to form thrombi. Thrombosis? I'm not sure which is plural. A thrombus is the technical name for a clot. And that process involves positive feedback, and it is triggered when there is a break in a blood vessel. A break in a blood vessel could be where you actually have blood that is at the surface of the skin, but it can also be something as simple as a bruise. Blood also helps to regulate body temperature. So the heat energy that is generated by the metabolism that takes place in skeletal muscle is distributed by blood. Blood contains a lot of different ions, which are usually referred to as electrolytes in this context. And so the blood is also important for the balance of fluids in different compartments of the body. And then blood pH is also regulated to some extent by blood. And that's essentially because there's certain blood vessels that have receptors that can detect hydrogen ions. And hydrogen ions, if you remember way back from chemistry, anything that has more hydrogen ions than hydroxide ions is going to be acidic. And then last, but certainly not least, blood also helps defend the body against disease-causing microbes, which we call pathogens. So when you look at blood, when you Blood's never blue. It only looks blue when you look at your light-skinned and you look, you see blood in your veins. It's not actually blue. That's a function of the absorption and reflection of light. When you actually take blood out of the body, you see that it has a range of red tones. So oxygen-rich blood is very bright red. Oxygen-poor blood, which is in the top, is more of a darker color. Blood pH is super tightly controlled between 7.35 and 7.45. The reason why, again, to remind you, the reason why pH is so important is that proteins have a specific pH that is that's optimal for their functioning. And if you get outside of that pH range, then the proteins denature. Temperature of blood is about 100.4 degrees Fahrenheit, and we have a fair amount of it. So adult men, 5 to 6 liters, and adult females, 4 to 5 liters. And those are rough ranges, right? You could have a smaller adult male and a larger adult female. I'm thinking some of the WNBA players that are really tall, they might have 6 liters of blood. So it's what we call an overlapping distribution. And blood makes up about 8% of our body mass. So if you pull a tube of blood out and you put it in a special tube that has a cap on it, and you pop it into a machine called a centrifuge, which spins that tube of blood really, really rapidly in a circle, what you see is that blood falls out, or you end up with sort of 3 layers to blood. And that's based on the heaviest parts of the blood fall to the bottom with the force generated by the centrifuge. So red blood cells, which are technically called erythrocytes, erythro means red, cyte is cell. So erythrocytes are about 45%. Then you have less than 1% of the volume of our blood is made up of leukocytes, which are white blood cells. Leuko means white, and platelets, which are involved in blood clotting. About 55% of our blood is plasma. And plasma is the ground substance. Remember that blood is a fluid connective tissue. So, and the definition of a connective tissue is widely scattered specialized cells that are embedded in an extracellular matrix, which is made of ground substance and protein fibers. So plasma is the ground. It's almost all water, and that water is absorbed primarily in the large intestine during the digestive process. So other than water, we've got big old proteins, which we'll talk about in a second, and then everything else. So we've got ions, which again, electrolytes, sodium, potassium, chloride, magnesium, calcium, small organic molecules, so amino acids, lipids, really that should say fatty acid, glucose, and then nitrogenous waste. Nitrogenous waste is just a fancy way of saying nitrogen containing waste. Then we've got trace elements and vitamins, right? The fat soluble vitamins are going to be hooked up to one of these transport protein that is, because if a vitamin is fat soluble, that means it's nonpolar and hydrophobic, so it's not going to dissolve in plasma. So if you want to move it around the body, which is what blood is for, is moving things, then you need to hook up things that are nonpolar to proteins that are soluble in water. And then gases. There are other gases other than oxygen and carbon dioxide, but I like to use those two in particular because hopefully it reminds people about aerobic cellular respiration. You have to transport oxygen because it's a reactant in all of our cells for aerobic respiration, and carbon dioxide is produced as a byproduct. Our bodies can't use the carbon or the oxygen in carbon dioxide, so it's purely a waste and has to be gotten rid of. So we got three major types of plasma proteins. You want to sort of know these three classes of proteins and which is the most common, which is the least common. So albumins are the most common form of protein, and there are a whole range of them. Albumins are often transport proteins for things like steroids and other kinds of lipids. So cholesterol, estrogen, testosterone, fat-soluble vitamins. The albumins are polar, and so they attract water. And because of that, the albumin content of your blood is important for determining what we call osmotic pressure. Essentially, how easy is it for your blood to hold on to water during this process of exchange across the smallest blood vessels. Then we have globulins, which are, you guessed it, sort of globe-shaped. Gamma globulins are kind of antibody that are really important in immune system function. Other globulins are important for transporting nonpolar materials and transporting iron. So we're going to talk more about hemoglobin, which is the globin that transports oxygen. And that's the one that is in our red blood cells. And then last we have a protein called fibrinogen. When you see O-gen at the end of something, you're told it's a protein. This is a sort of a fibrinogen it's a protein, but it's in its inactive form. You actually need the presence of calcium in order for the enzymes that chop fibrinogen into the pieces that are to create the active form of the protein, which is fibrin. And that's absolutely critical for forming blood clots. All of these proteins are produced by our livers. So it's another, we tend to think of the liver it's just about bile, but it's also about detox and making all of these different plasma proteins as well. Okay, so there are three different formed elements of the blood. And so we've got erythrocytes, which are red blood cells. And this is from a blood smear. So things are more or less in a single layer. There's a little overlap in some cases. And what looks like a hole in the middle of these red blood cells is actually the concavity. Up in the upper right corner, what we're looking at is a red blood cell, an activated platelet, and then a white blood cell, a leukocyte that's been taken with an electron microscope and then colored to make them stand out because electron microscopes only give you black and white pictures. So white blood cells, lots of different kinds of them, lots of different kinds of leukocytes. And we'll learn a lot more about leukocytes when we talk about the immune system in, I think it's week 12. And then platelets. Platelets are sometimes called thrombocytes because thrombus is clot, but they're not actually cells. I always try to use the term platelets. They're actually little blebs off of these huge cells called megakaryocytes that are found primarily in bone marrow, a little bit in the lungs. And it turns out one of the weird things they've recently found out about being seriously ill with COVID-19 is that upon autopsy, they're finding these megakaryocytes in the brain and in heart, which is just unheard of. Okay, so hematopoiesis. If we were in class together, I would make you guys say it with me. So heme means blood, and poio is Greek for make or create. And you have the ATO as a joining to join the two together. So hematopoiesis is the process of creating blood. And this is what you need to remember for this week about the process of hematopoiesis. When we get to the immune system, it'll make sense to remember more, but I want to introduce you guys to it to start with. So it happens in red bone marrow, right? The younger you are, the more red marrow you have. If you're an adult, the only place you have red marrow in terms of your long bones is going to be in the epiphyses of the long bones. All of the formed elements come from a multipotent stem cell. It's called a hemoblastocyst. You don't need to remember that name, but and then the hemoblastocyst, when it divides, it gives rise to another hemoblastocyst and either another kind of stem cell. These are all stem cells thus far, called a myeloid progenitor. So a progenitor is sort of, it's like an ancestor in a way. If I'm a red blood cell, I share a common myeloid progenitor with the megakaryocyte, for example. But you've got these two main stem cell types after the hemoblastocyst, either the myeloid line or the lymphoid line. One of the reasons why I started talking about this earlier in the course is usually somebody would ask me about leukemia, because there's myeloid leukemia and then there's lymphoid leukemia. Part of the way they're diagnosed is by looking at blood and bone marrow to see the relative proportions of myeloid and lymphoid progenitors and cells they give rise to. So in the myeloid line, we've got the megakaryocyte, which is going to give us the thrombocytes / platelets, erythrocytes. Everything else on this page, these are all different kinds of white blood cells. So we'll get into that in more detail when we talk about the immune system, when it makes sense to do that. So as I said, erythrocytes are called or red blood cells are called erythrocytes, and they're described as biconcave discs. These cells are so full of hemoglobin proteins, which is the protein that transports oxygen, that they actually spit out the nucleus as they mature. And what that does, by having this biconcave shape, you end up with more cell membrane for gas exchange. As you might have guessed from looking at that blood smear, we have lots and lots of red blood cells. It's the most common formed element in our blood. And by the way, a microliter is 1/1,000,000th of a liter. So, if you figure out sort of whether you think you have 4, 5, or 6 liters of blood, depending on your size, multiply that by 1,000,000 and then multiply it by 4 or 6 to figure out how many red blood cells are floating around. Erythrocytes primarily contain this pigment called hemoglobin. It's specialized for transporting oxygen. So the hemoglobin protein has quaternary structure. So remember, there are 4 levels of protein structure. We've got the sequence of amino acids, which is the primary structure, then the secondary level of structure, which has to do with alpha helices, which you see a lot of in hemoglobin, or the beta pleated sheet. Tertiary has to do with the R groups interacting. And then quaternary is when you have multiple proteins coming together to make a super protein. So hemoglobin is a super protein. It's made of 4 globin proteins, and each one has a what's called a heme group. That's green in this image. So if we look at that in a little more detail, it actually is a what's referred to as a planar molecule. So the heme group really is flat, and sitting right in the middle of it is an iron atom. And capital F, lowercase e is the symbol for iron in the periodic table. So each iron can bind a single oxygen molecule, and it does that reversibly. So it goes from deoxyhemoglobin to oxyhemoglobin, right? The D means the oxygen has popped off of the heme group, and that happens based on the concentration of oxygen where you are in the body. And that's a really important point, because there are differences in oxygen content. For example, in the small blood vessels in the lungs, the reason you send blood to the lungs is to pick up oxygen, get rid of carbon dioxide. So there's not a lot of it attached to hemoglobin in the lungs. If you go often look at oxygen content in the tissue fluid in your big toe, your cells have been doing aerobic cellular respiration, using up the oxygen. And so then there's a low level of oxygen in the tissue fluid and a high level of carbon dioxide. There's a lot of oxygen and little carbon dioxide in the blood. So we're going to be talking about cell transport again, because oxygen, gas exchange in our body is driven by diffusion. Okay, so, right, if you remember aerobic cellular respiration, you've got glucose plus oxygen yields carbon dioxide and water, right? You need 6 oxygen molecules to completely combust or burn a single glucose. If you have incomplete combustion, so you're burning wood in a fireplace and there's a lot of buildup of material in the chimney, and your house is really airtight, you can end up with carbon monoxide, which is CO. Carbon monoxide is incredibly dangerous because it binds to hemoglobin just like oxygen does, but the binding is irreversible. So once the hemoglobin is saturated with carbon monoxide, it's not useful anymore. You essentially have to wait until you get more red blood cells, which is why carbon monoxide poisoning is so dangerous. It's also the case that carbon monoxide is many times more attractive to hemoglobin and vice versa than oxygen is. So essentially carbon monoxide, if it's in your system, is going to outcompete oxygen molecules for access to transport by hemoglobin. And this is my dumb science joke, which I didn't actually make. I just got a kick out of it. And it does kind of make the point. You know, hemoglobin is supposed to be transporting oxygen, but it's a lot more attracted to carbon monoxide. Okay, so erythrocytes produced in red bone marrow. As I said, they spit out the nucleus as they mature. And so we're looking at some spongy bone here, up close and personal, a bunch of red bone marrow, and then some small blood vessels that are going through the red bone marrow. You can sort of make out the erythrocytes there. All of the other cells that are a little darker colored are different kinds of either white blood cells or different stages in the process of making blood or making the formed elements. Erythrocyte production is regulated by a hormone called EPO, which is short for erythropoietin. So, right, if poio means make or create, erythropoiesis is making red blood cells. Erythropoietin is a molecule that stimulates red blood cell production. So, right, if poio means make or create, erythropoiesis is making red blood cells. Erythropoietin is a molecule that stimulates red blood cell production. It's a favorite for blood doping by endurance athletes because the more red blood cells you have, the more oxygen you can carry. Unfortunately, it also makes your blood a lot thicker if you have a lot more viscous, if you have a lot more red blood cells. So you're at much higher risk for clotting in places where you don't want to have clots. It was originally developed for folks who are having chemotherapy because chemotherapy targets fast dividing cells. It's a blunt instrument, and the side effects of traditional chemotherapy are like we lose your hair, you stomach upset, right, and you have dry, itchy skin. All of those are tissues that turn over really fast, so high levels of cell division. Same thing's true with bone marrow. And so the fatigue that can accompany chemotherapy is because you don't have enough red blood cells to transport oxygen, and if you don't have the oxygen, you can't recharge your ADP into ATP. So red blood cell breakdown. So these guys, right, they don't have a nucleus, which means that they don't, by definition, they can't have a very long lifespan. 3 months, no more. And then the spleen and the liver start to pull them out of circulation. There are phagocytes, which are white blood cells that are present in both the liver and the spleen, and they are sort of constantly checking for aging and sort of past the expiration date red blood cells. So they will phagocytose them, and then they reuse a lot of the components they find in the red blood cells, but they also then break hemoglobin down into the globin chains, which are broken down into amino acids and then used by the body. And then the heme group. The heme group is split in the liver into iron and a molecule called bilirubin. So the iron gets transported to bone marrow, where it's used to make more red blood cells, and the bilirubin the liver converts into bile, which is then stored in the gallbladder. Big things to remember from this slide is how short-lived red blood cells are, erythrocytes are, that they are pulled out of circulation by the spleen and the liver, and that their components are repurposed and reused. This video, I play it in part because it's old. I think it's kind of groovy, but also it really makes the point that this process of diffusion, which is passive transport, which remember is based on concentration gradient. So oxygen is going to move from where there's more of it to where it's less, and the same thing is true with carbon dioxide.
One drop of blood carries millions of these cells, the red blood cells. They have this distinctive shape, which gives them a large surface area to absorb oxygen, and no nucleus, so they have maximum room for the substance hemoglobin. Hemoglobin is a bright red pigment and gives blood its colour. It's a globular protein with the metal iron at its centre. It's the presence of the iron that gives hemoglobin its oxygen binding and releasing properties. The hemoglobin is transported along the blood vessels within the red blood cells.
So how does hemoglobin work? How does it know when to pick up oxygen and when to let it go? Well, in areas where oxygen concentrations are high, such as in the lungs, oxygen passes into the cells and binds loosely with hemoglobin to form oxyhemoglobin. In this state, it travels through the body until it reaches an area where oxygen concentrations are low, such as close to working cells.
In the working tissues, the red blood cells let go of the oxygen and revert back to hemoglobin, ready to be used again.
So we'll circle back to that when we are talking about the respiratory system. So platelets are sometimes called thrombocytes. They are the result of little blebs, little bits of cytoplasm with a little bit of membrane coming off of these ginormous cells called megakaryocytes. Again, made in the red bone marrow. We make lots of them every single day, 200 billion. And we have, depending on your particular genetic makeup, 150 to 300,000 per 1/1,000,000th of a liter. And those guys are the ones involved in blood clotting. When blood vessels are damaged, that's the stimulus for blood clot formation. And in order for a clot to form, you need platelets, you need roughly 12 different proteins that are clotting factors, and calcium ions. One of the clotting factors that you absolutely have to have is, so platelets, when there's no break in the blood vessels, bounce around inside our blood vessels, and you can imagine, right, if they were in their activated state where they have all these sort of spikes, they would start to stick to things, which is exactly what happens when a clot forms. So the clots are essentially fibrin, mesh, and platelets, which then start to trap red blood cells, and white blood cells race in because a broken blood vessel stimulates inflammation.